Hi and welcome to today's webinar, 5 Steps to Clinical Relevance. Thank you for taking the time out and being with us here today. We'll be running a live Q&A at the end of the webinar, so we have enabled our Ask a Question feature, and you find it on the right-hand side of your screen. So if you have any questions along the way, please pop them in there. And if you miss anything, don't worry about that, because we will send out the link to the recording afterwards. My name is Sofia Erling, and I'm the Business Development Manager of Immunid, and I will monitor today's webinar. We will talk for about 30 minutes, and then we have 10 minutes for your questions at the end of the webinar. Our panelists that are with us today is Justina Leija Jarblad. She's the Chief Operating Officer of Immunid, and also Bengt Dahlström, Chief Scientific Officer at Clinical Trial Consultants. We are two companies based in Uppsala in Sweden who work in close partnership, providing preclinical safety testing and conduct clinical trials. So a short introduction to our panelists before I hand it over to them. Justina, she has extensive experience within oncology and virology, as well as from drug development. She has worked for many years with preclinical development and first-in-human clinical studies. She is also the co-founder of the first successful crowdfunding campaign called iCancer. This is to support drug development of virus-based drugs. Our other panelist, Bengt Dahlström, he is Associate Professor in Pharmacogenetics and Drug Therapy. His expertise lies within pharmacogenetics, drug research and development, where he has over 40 years of experience from leading positions in pharmaceutical companies and CROs. And with no further ado, let's look into the first step to clinical relevance, guidelines. They can help in the selection of the most appropriate test, but they do not state exactly which tests need to be carried out. Guidelines are constantly updated, and Justina and Bengt will tell you more about how to relate to guidelines and how to mitigate the risk for the healthy volunteers. So I leave the word over to you, Justina.
Perfect, thank you. So in regards to the guidelines, focus lies to support development of biologics, which is a very complex, large group of drugs, everything from small peptides to large cells that can be used as therapies. And biologics or biotherapeutics can interact with the human body in many different ways, especially on our immune system. Therefore, guidelines do focus right now on the aspect of biologics and the capability to trigger immune toxicity or immunogenicity. So assessing the potential of the drug to increase or decrease activity of the immune system became a very important part of the preclinical development for biologic. And safety evaluation should include both intended, so pharmacological action of the drug, as well as unintended, so toxicological action on the drug on the immune system. And among other things, the regulatory agencies highlighted the importance to use human-based models in order to study the side effects or the immune adverse side effects that can be caused by biologics. This can be done by the use of the human whole blood or peripheral blood mononuclear cells, so-called PBMCs. And the emphasis to use human-based models came clearly from the TGN 1412 incident that caused a severe cytokine storm in human subject. This immune-related side effect was not predicted in the animal model, that was a primate, but was later established on the human-based model using PBMCs. However, guidelines cannot determine what human model or assay can be optimal for each individual drug. And as I mentioned, the use of human cells can be actually applied by different protocols, different assays. We can use so-called high-density PBMCs in aqueous phase or solid phase. We can use whole blood on plate or whole blood loop system So there are a variety of assays and methods that can be applied in order to actually include human-based data. And it is up to the sponsor to determine what immunological assay is the optimal for your particular drug. So this cannot, this responsibility cannot be put on the regulatory agencies. It is actually the responsibility of the sponsor. And keeping that in mind, I would like to emphasize one important aspect when you're working with the service providers. So I have encountered on different websites messages that can be read like this, and I quote, PBMCs is one assay recommended by the FDA. This statement is correct. PBMCs is recommended as an assay, as a human-based model. However, it's not the only assay that is recommended, and often it's the assay that might not apply to all different biologics. So as an example, the case TGN-1412, the cytokine release against this antibody was proven in the assay that was called PBMCs with the solid phase. However, the same assay was proved to be not effective to show cytokine release against antibodies like rituximab or alemtuzumab that are known to actually trigger cytokine release in the clinic. So this is very important to keep in mind that there's no standard in regards to what human model to use. So the key messages from me in regards to this first point is that the human models are very important. They should be included in the preclinical development. However, there is no standard, and the sponsor has the responsibility to choose the assay and provide data that support safety profile of the particular drug. So with that in mind, I will now leave the word to Bengt.
Yes, and I will say a few words about the clinical side. When talking about first-in-human studies, the safety for the volunteers is the primary concern. And to mitigate the risk for the volunteers, we need information of the type that Justina just mentioned. We need information on receptor binding, Mabel, NOAEL niveaus, PK and TK in animals. And with all this information, we do an allometric scaling to arrive to a human equivalent dose, the dose that we can use in humans. To this, we will apply a certain safety margin and determine the starting dose for the compound. And as Justina said, it's a great benefit if we have ex vivo data, which we also can rely on, not only on the animal data. As always, the amount of information varies between different projects, and lack of preclinical data usually makes our risk assessment, the clinical risk assessment, more difficult. And with the lack of information, that usually translates into a more careful approach when we determine, for example, the starting dose, the safety margin, the dose escalation steps, and the dosing intervals. And especially talking about dosing intervals, the time between dosing two individuals in a study is something that we based on preclinical data on both pharmacokinetics and toxicokinetic data. And their time courses, of course, carry very important information on how long we should wait and how long we should follow with observations after we dose, and also how frequent we can dose with the INP. This is a very important safety aspect, that we don't dose too many subjects at the same time, which if everything goes wrong, we will have a lot of problems occurring at the same time. So this was my comment on the clinical side. And now over to Justina.
Sophia, I think we are in the second point now.
Yes, sorry, I must have a glitch with the sound there. But thank you, Bengt and Justina. So over to the next step. Instead of asking which test will meet the guidelines, you should ask which tests are relevant for your drug candidate, but it's not as obvious as it sounds. Now, Justina, you will share some first-hand experience on how to determine what's relevant and what's not.
Yes, thank you. I have been working a lot with different projects at the preclinical stage, and in general, regardless of the project, the regulatory agencies do support the principle of the 3 R's, so replace, reduce, and refine, of animal use in the preclinical testing if it's feasible. I think that the questions that are important is rather not that if animal models are bad or good, it's rather what animal species might be relevant, if they should be used, or how many species we should be including in order to provide the data that might be relevant for the drug. And again, we go back to the sponsor responsibility to choose the assays. And I would recommend not to apply methods or assays used in the previous pipelines or project, but to think from the start, since usually the drugs do change in the structure, they do change in their function, in their targets, and therefore the methods or the assays also should be re-evaluated. If we think about the in vitro models and how to use them, apart from the relevance, diversity always will build the value. So as an example, if you intended to use the drug intravenously with the aim that the drug should act in the tumor microenvironment, do include model that will somehow represent the journey of the drug in the body. Look at the model that can build all the interaction of the drug with the human blood, even if you don't have intention to have any effect on the blood cells. And also then include models that can represent how the drug can behave in the tumor microenvironment. So if the drug is intended to use intravenously, it is also of importance to look at all the different blood components that are present. So I would recommend to think more which models are the most physiologically relevant. If we think about the safety evaluation, usually it refers to the cytokine release assay as a sign of cellular activation. However, in the blood, other components can trigger infusion reactions or hypersensitivity reactions. For example, complement. So some drugs can act through complement as a part of the design. For example, antibodies can act through the CDC, the complement dependent cytotoxicity. And some drugs might trigger complement activation unintentionally. This can be a part of drugs based on liposomes, nanoparticles, or oligonucleotides. So I would then think about looking at, when studying different interactions of compartment of human model, if we do it separately, one thing we need to keep in mind that we might miss how those different compartments or interactions will work together. And therefore we might diminish relevance of our results if we only focus on the separate assays. Therefore, my main message here would be to build safety evaluation on as many readouts as possible. So diversify your in vitro assays, include cytokine release, complement activation, platelet activation, since that's important part of the blood environment, which might be important for all the drugs that actually are administered intravenously, but also increase the relevance of your results by trying to apply as many readouts as possible in the same sample, because then you can study different interactions in the same network which will actually give you a better picture of what can happen when your drug is infused in the human body for the first time. Now I will leave word to Bengt.
Yeah, and I will just follow up a little bit what you said about the complement system, because this, of course, is a very important part of the innate immune system. And it has, one of its primary functions is the defense against infections. And this means that it will or can influence the study design in such a way that if you have increased activity, which can lead to a too powerful immune response, you need to consider whether are there any antidotes available? Do you have other rescue medication which can dampen the immune system effect? Or in a case where you have decreased activity, meaning that the individual could be more susceptible to infections, meaning that they might have to be isolated during the period while the immune system is depressed. They might require vaccination against certain type of bacteria, or they could use pre-medication like with antibiotic, for example. And one example is the meningitis, which is a serious infection that could occur if you suppress the complement system. Yes, over to you.
Okay. Thank you. So I just want to remind you to write your questions in the Q&A and we'll take them at the end of the webinar. We're halfway through now. So the fourth step to clinical relevance, confirm the negative. The absence of toxicity in the preclinical stage is no guarantee that side effects won't show up in the clinic. In fact, from the clinical perspective, it's not that uncommon to see infusion reactions. And now we will talk more about one of the most infamous examples of this.
Yes. I think you all know the case of the TGN-1412, where, which I think still is very good, a relevant case to actually prove the principle that we want to talk to you today about proving the negative. So in this case, we have a promising preclinical testing that was done on primates, macaques, that show very nice safety profile. And then the dose of TGN-1412 was that was used in the human subjects was actually 500 times lower than in primates. And even those safety concerns caused that TGN-1412 actually caused a severe cytokine storm and organ failure in human subjects, regardless the testing in primates and the lower dose. So while primates are very similar to humans, the expression of one surface receptor different between the two species. And it was very interesting to know that actually the signs of activation was seen during studies on rodents. However, the rodent model was deemed to be far simpler or not that relevant or not that related to humans. But in this case, the rodents were a more relevant model than primates. However, they were ignored in the favor of the obvious primate analogy. So regulators have questioned the predictive model of animal testing when testing for a new biotherapeutics. And therefore, there was the requirement to use the human based models as well in order to confirm if the cellular activation seen or not seen in animal testing can be also applied to the human situation. So we read in the guidelines that signs of cellular activation in vitro should always be taken into consideration regardless of the negative findings from animal studies. So for the TGN-1412, it was this unfortunate case when a more advanced or model was chosen over the simpler but relevant model. It's interesting to know that TGN-1412, it is currently back in the clinical development right now under the name TAB-08. And of course, it was applied in a much lower dose, and so far there was no severe side effects observed as it was with this first clinical trial. also in, as an example, the Bali trial, we see again the more advanced model, I think it was dog model in this case, failed to show neurotoxicity in the preclinical setting. And it was proposed that a much simpler in vitro cell-based assay could have provided the information about the side effects that were observed during clinical trial. So it's the main message here would be to always confirm the negative by adding additional tests. And another example in regards to the biotherapeutics could be if you work with the checkpoint inhibitors and your main focus is T cells as your effector cells, it's always good to remember to look at the assays that maybe include different cell types as well. In case the target might be expressed on other cell types, for example, neutrophils, if you use only PBMC assays where those particular cells are not present because they have been isolating away in the process, you might not see the complete picture or the complete safety profile of your particular drug. If you have interest to study antibodies that should act through both ADCC or CDC, then the whole blood assays might be preferable in your case. It might go without saying that the cost should be always secondary. In the preclinical stage, I think that if you apply the idea about to diversify, but also keep the relevance in mind, you might actually minimize the cost by choosing the assays when you combine many different readouts in one assay rather than applying many different methods in the same time. So the bottom line is relevance, not complexity. And the main goal is not to show negative results. The main goal is to prepare for any possible side effects that might happen in the clinical situation and predict risk of immune-related adverse events. And Bengt, now.
Yes, I'm sorry I dropped out for unknown technical reasons here.
Oh.
But let me say a few words about the infusion reactions. Of course, as Justina mentioned, they are not uncommon in a clinical perspective. And they can be both local and they can be systemic. They can be immediate, like the anaphylactic shock, or delayed. We must always be prepared for them in the clinic for them to happen and have the appropriate resources available, meaning you should perform this kind of studies in a first-in-human setting. You should have staff with experience from both first-in-human studies and intensive care. And you should always have a backup from an intensive care unit on call when you do this type of studies. Now, whether we can tolerate them or not, that really depends on the risk-benefit balance, meaning that a more risk can be tolerated for example, life-saving drugs for serious diseases. While for other types where the effect is not that obvious, you might not be able to take more risk like that. It's also very important to know a little bit more about the type and the duration of the immune reaction, and if there are any antidotes available, or whether you can use pre-medication or co-medication to dampen the immune effects. A simple measure like infusion rate can also be used. If you lower the infusion rates, it usually means that you will get a less intensive immune response. So that could be one way to circumvent the problem. It's also important to, perhaps in certain cases, to implement a certain period where you supervise the patients after the infusion. This is not uncommon when you use biological antibodies, for example, anti-inflammatory antibodies, that you will have surveillance of the patient for a certain number of hours after the injection, just to make sure that you don't get any immune reactions during that period. The big question is really, is this a showstopper or not if you get a side effect like this? Well, it really depends on. I would say no, probably not, if there's a high unmet medical need for the drug and the immune response can be kept under control, for example, with pre or co-medication. In other instances, I would say maybe not. For example, if you don't know so much about the clinical efficacy or the clinical response of your drugs because you're so early in your drug development program, immune response that leads to serious adverse effect that cannot be counteracted, then there might be reason to put the question to yourself, if you're the sponsor, maybe it should be better to redesign the drug if that's possible, or do some other changes, meaning that you could avoid this, because it's very costly to make failures later in your drug development program. God forbid under phase 2 or 3, then it will have huge cost. The cost if you do something early on is usually very much lower and is easily to defend. And it could also mean that your whole time course during the drug development program will actually go faster if you take this step back early on. Yes, thank you.
Okay, thank you. So if asking which tests are expected is the wrong question, what are the right questions? Justina and Bengt will now outline 4 important considerations to keep in mind when approaching the clinical phase.
Yes. Many times actually, when you do select your lead candidate and you go for the production phase, that is a very expensive commitment. Therefore, I would always recommend to considering moving some of the in vitro testing a little bit at the early stage, rather than to wait and the late preclinical stage when there is a new investment in the form of GLP toxicity testing. And when you select your in vitro assessment, always think with the endpoint in mind. And I have listed some examples that might be relevant here. So again, a lot of biotherapeutics are actually used intravenously. So then checking how the drug and the formulation buffer selected for that particular drug should be one of the very early tests you can do. How it behaves in the blood. One example, it's very simple, fast, and cheap assay. That's a hemolysis test that actually I have noted sometimes is not included in the preclinical testing. And this is just a simple assay to evaluate if your drug formulated in the buffer that was selected can affect or have a lytic effect on the red blood cells. And it's in terms of the formulation buffers, their role is to make sure that your drug is stable and do not precipitate. However, it is good to keep in mind that once the drug actually meets the blood or the plasma, it might behave differently. It might precipitate, aggregate, and then cause an activation that maybe was not intended. So actually looking at checking the environment you are actually intended to use your drug can be very, very simple and easy assay to be included already in the post-production stage. Also, to therapeutics, usually it's quite complex process. And for example, in the production stage, you might have a purification or other processes that can be optimized or modified in order to increase the quality of your end product. And here I found that in vitro testing, looking, for example, for monocyte activation, is a very simple way to determine which purification process, for example, can be optimal and give you product with the highest quality. And this is, of course, in vitro assays that are adjusted to look at endotoxin content or any debris from the cells like DNA that might cause unspecific activation if it's present. I think that based on the projects we have been involved with, I think it's very important to don't wait with the complete safety profile to be done at the late stage of the preclinical development. Many assays can be already applied and done very early at the production stage that can actually give you a better understanding how your drug might behave later on. Bengt?
Thank you, Tina. You gave some very good examples here, and let me just take one more that is sometimes overlooked because it's really so simple, and that's the influence of the infusion devices. Many biological, as you said, are administered as intravenous solutions, usually in low concentrations. And protein and peptides are, of course, known to be absorbed or adsorbed to the material in the infusion devices. And it's important not only to look at the cannulas or catheters, you should investigate how the syringes behave, the valves, the catheters, and it's both a question of material and the volume that you will use, and also in the case of catheters, the length of the catheters. So different materials behave differently, so it's important to test the specific device that you're going to use in a certain study. And some materials are known to be less absorptive than others and could be used, but it's very much dependent on the characteristics of your molecule. So besides the material, of course, it can be influenced by the pH of the solution, whether the molecule is hydrophobic or hydrophilic at its surface, the concentration, of course, and also other ingredients which could be in the solution that could affect this. So it's fairly simple and straightforward, but usually this type of test is usually performing rather close to the start of the clinical part, and it's so simple and it should really have been tested long before that. And especially when it comes to the things like aggregation behavior you mentioned, Justina, that's also very important, because what we're doing when we are infusing, we usually get very high concentrations of the molecule at the injection site, and it could mean that you will have buildup of aggregates, for example, and not all molecules are that prone to form aggregates. So that's another thing that you really should investigate, because you have self-aggregation of certain type of compound. And you mentioned also that this could be one factor that could influence the immune response to a big aggregate, for example. Yeah, so those were just a few examples.
All right, thank you. I see that we have some questions here, so let's take the final step before we head over to the questions. So last step, fast does not mean first. We all want a success story, just as we all want to move quickly and make money from our innovation. But taking time to ask the right questions at the preclinical stage can actually speed up the whole process. Bengt, please share with us your final thoughts on how to move from preclinical testing to first-in-human trials in the best possible way.
Well, this is a 100,000-pound question, really, what is wrong and what is right. But it is important to always try to take informed decisions based on the best available knowledge that you have. So we need to remember that drug development is a timely and very costly endeavor, and it's associated with a very high risk for failure. Anything between 5 to 15% of all started drug development projects never really reaches the market stage. And it's also important to remember there is no single cookbook recipe to follow, but you need to tailor make each and every product that you enter into in order to help the sponsor to build a cost-effective development program. This is why a risk-based approach is so important. That is that you need to pay attention to all the details, but at the same time never lose oversight where you're going. So it's important not only to do the right thing, but also to do the right thing right. So in a sense, I mean, you could do everything according to the book, but if the book doesn't tell you the relevant thing from a scientific point of view, it might not be worthwhile doing it from the really start. We should always remember that some shortcuts suggested to us may actually turn into very costly delays if we don't do the right things. Drug development is a teamwork between a number of different competencies, and at its best, it's really the foundation for a successful drug development. It's not a one-man show, but really it's a team effort that will bring a successful drug development program together. So there are, of course, challenges. There is always a financial component to most of the drug development programs run by smaller companies. You need to have investors investing in your idea, and they are very keen on making sure that you adhere to agreed timelines and milestones, etc. And there's usually very little slack time in a product plan like that. But things can happen during the way, and you also need to make sure that you have taken that into account when you do the planning. Things can go wrong. You need to go back a step in order to get forward again. It doesn't mean that you will actually, in the final end, will lose time, but you can actually gain also time from doing it correctly from the beginning. It's also important to remember that what we're trying to do is to help our client, our sponsors, to build value into their product, because that's the guarantee for them to be able to raise more funding to continue the project. So that's why I say it's seldom that you have a cookbook recipe that you can follow. You need to design each and every product according to what actually builds value in the product they are involved in. And this is also what their shareholders will expect to see from you. So with those few remarks at the end, I would like to thank you for listening to this seminar, and we look forward to the questions that you might have after this. Thank you so much.
Yeah, thank you so much.
Okay, thank you, Bengt and Justina. We will go ahead and take some questions now. And just a reminder, be sure to type in your questions in the question box. And all your questions are anonymous, so just ask away. And it looks like we have a few questions. So I will start with the first one. It says, hi, the TGN-1412 side effects on humans could have been predicted if there was more preclinical in vitro assays. Justina, maybe you would like to answer that one.
Yes, sure. With the TGN-1412 case that was so well studied is that there was use of the cells in aqueous format prior the study, and there was use of the different animal models. And again, in the post era of TGN-1412, it was shown that this kind of differences between the human and the animal caused that the primate model was not effective, and that the drug require cross-linking by simply air drying of the antibody on the plate in order to create the assay that could be truly predictive. So I think that the part of the TGN-1412 story is that there was a bit of lack of diversity, or that basically a negative results from the primates were the major findings that were used to push the drug forward. Because that's, again, you do have the sign of the activation from the drug in the rodent studies. So I think that this case changed the way we look at the preclinical and clinical development, in the sense that right now animal studies can give you a certain idea about the behavior of the drug. However, if you would see any signs of activation from the human model will be actually taken into consideration over the animal studies. As I mentioned, knowing the structure and the the end point, how you want to use your antibody in the human subject, can help you a lot to choose the assay that will be more relevant. So, as an example, if your antibody or require cross-linking, then you have to apply assay that allow to study that. If you want to act through ADCC, then of course you need to have NK cells. If you want to have CDC effect, you need to have a active complement system. Regarding what are the structure of your product, it can be a protein. If it's recombinant, then of course you need to have a look at immunogenicity, but also you can have a look and if there's any interactions with the complement or clotting system. So I think that we learn a lot based on the TGN 1412 case. And I think that all the tips and comments we had today are a very good summary on how you should think in regards to what assay to choose to prove the safety profile of the compound.
Okay, thank you, Justina. And I just would like to point out that if you have additional questions, you are happy to just send us an email, and we will get back to you as soon as possible. Another question that is for you, Bengt. Important items to consider when manufacturing the drug product to be used in first-in-human studies, or maybe you, Justina, you can choose who would like to answer that one.
Well, I can maybe start there. I was thinking here that from the practical point of view, it's very important to know what does the drug or the final drug product look like when it comes to the clinic. Usually when we are working with first-in-human studies, that's not a fixed dose, but several different fixed doses which we are going to use. And it's very important to think about the practicalities, because it's very seldom that the sponsor can deliver all the different strengths to the clinic, because we really don't know from the beginning which one we will use. So usually it means that we will often get some kind of stock solution which we need to dilute. And we can do that extemporaneously at the clinic in certain cases when there is short half-life and you're going to use the product immediately. But it has to be well thought through what the concentration of this stock solution will be. So we won't have too many dilution steps, because that will increase the risk for human errors or mistake, that something will go wrong in the dilution scheme. So that's one important aspect, of course. Then the other thing, which has happened more than one time, is that you need to think about the batch size. Don't forget that it should not only be enough material to run all the patients that you plan, but it should also be stable for the period that you're going to run. In many instances, at the end of the study, we have reached a situation where the shelf life has run out and the company has to reapply for extension, etc., which can take some time to get in place, everyone. So it's important not to underestimate the amount of drug that is necessary for the clinical trials. Another strength is, of course, if you use the same kind of production batch for the animal as for the human studies. That's just a few comments on that.
Okay, thank you. Another question that we have is, can we study anything else but PK and safety after dose escalation in a first-in-human study?
Well, maybe I should say a few words about, yes, definitely you can. Don't forget that first-in-human studies are exploratory studies run in a limited number of subjects. It's always adaptive to its design, meaning that we will take decisions based on the findings during the study to steer the rest of the study. The best example is, of course, the different dose levels. We always evaluate each dose level, whether it's safe or not to continue with dose escalation. That's one adaptive decision as is taking place during these types of studies. But since it's exploratory, you have the luxury of actually start looking into other areas. And it's very common to have a single cohort looking at food effects. Now, why is that so important? Well, when you do the single ascending doses, you usually work with healthy volunteers which are fasted. And when you turn over to multiple dosing in volunteers or in patients, for practical reasons, it means that it will be taken together with meals. And you need to have a first rough understanding if there are any big food effects, because that means that you might have to adjust the dose regimen, for example, if you have big food effects. The other thing is healthy volunteers are usually younger persons. And when you start doing your clinical studies in phase 2, you usually work with elderly people, and there can be differences not only in the pharmacokinetic, but also in the pharmacodynamics. And you have differences due to concomitant medications. These are typical things that also can be investigated as a small cohort in the first in human study. Another study which is also very often added, that's a thorough QT part, not really the big one, but what we call an expert QT studies, meaning that you can start looking at heart effects already in the first in human study. And again, to get the first feeling for whether this is a no problem or if there's a problem that requires further attention in your clinical development program. So these are just a few things. You can look at drug-drug interactions, for example. You can look at mass balance, drug metabolism, etc. So there are a number of things which can be very handily built into the first in human program.
Yes, I can add to refer to what you said, Bengt, that one important aspect of the clinical trials is also to maybe apply or work with the biomarkers that can give you a better understanding how your drug behaves. And some of the work you can actually really start preclinically in regards to working with the healthy volunteers, if this is the cohorts that will be included in the first in human. But you can also include or sample individuals with a certain disease background to understand and compare those groups side by side in the preclinical assays as well, to understand how the drug behaves in these two different environments. And this kind of comparison might be also very valuable to establish what biomarkers might be required or may be needed for the clinical phase. That can be also included rather early as an exploratory point that can also build the value for late trials.
Absolutely.
Okay, thank you, everyone. I think we need to wrap up now. Can you hear me?
Yes.
Hello?
Hello?
Yes. You're online, Sophia.
I'm not sure if you can hear me, but I will try to wrap it up. Thanks everyone for being here today, and we are happy to tell you more about how we help clients with preclinical and clinical services. So don't hesitate to contact us with any questions that you have, that you might have. Thanks again, and see you next time.
Thank you so much.
Thank you so much. Bye-bye. Have a nice